So I'm really grateful to be presenting today. I'm grateful for the opportunity. I've been a fan of BAGIM for a long time, and unfortunately haven't been able to be in the Boston area that much. And with moving things online, I'm really glad to be able to talk to everybody. Please feel free to turn on your video. I like to have a little bit of an audience. It helps me gauge sort of what's going on with the presentation. But as Brian mentioned, I'm working at Google now, and I'm on a team in Google Research called the Google Applied Science team. And there's lots of different subgroups within that organization, but I help to lead a team that is focusing primarily on applications of machine learning and Google technologies to the drug discovery process, in particular preclinical work with hit lead and lead optimization. And my plan for today is to talk about a few different topics. Yeah, and as Brian mentioned, please ask questions in the chat along the way. We'll get to them at the end. There's so many people on that we probably can't stop and have live questions while I'm presenting. But this picture here is me in about 2006. I was a missionary for a couple years in West Texas, and they didn't mince words with the way that they spoke or the way that they named their towns. I lived in a place called Brownfield for a while, Levelland, Plainview, and I hope to be equally plain as we talk today about these three different things. So first, we're going to spend some time talking about prospective validation. This is based on an article I wrote for Trends in Chemistry last summer, and where I get to talk about some of the ways that we think about validating our models. Then I'm going to spend some time talking about work that our team has done in collaboration with XChem and Zebi AI on applying machine learning to DNA-encoded libraries for hit finding. And finally, I want to talk about an effort that Connor Coley and I are heavily involved in called the Open Reaction Database. And I hope that as I present on these three topics that may seem fairly disjointed, that you'll see common threads in terms of both gathering data and using it effectively to validate and improve the models that we're building today for molecular property prediction and lots of other interesting tasks. And then I'll leave plenty of time at the end for questions so that we can get some feedback from all of you that have been kind enough to spend your time with us this morning or this afternoon. So first, let's talk about prospective validation. We sort of all know that when we build models and we use historical data in a retrospective sense to validate our models, we know that that's not the best. It's a common refrain that, well, what I'd really like to do is apply this prospectively, but I don't think we talk enough about the reasons why it's important to work prospectively and some of the specific failure modes that retrospective validation can have. And so to frame this conversation, I want to start with this picture here, which is a sort of idealization of the design, make, test cycle that a lot of us go through in various stages of the drug discovery process. And many of the examples I'm going to give in this talk are focused on things like hit finding or lead optimization, but I want to make sure that it's clear that these processes can be applied in the abstract to any sort of iterative optimization. You could even apply it outside of drug discovery to things like materials design or whatever the case may be, where you have a sequence of events where you're working with building models, generating ideas, making things to test, and then evaluating them. By the way, when I move my mouse, does it show up on the screen? Yes, great.
It does.
Okay. So here is a further abstraction of that cycle that I just showed, where I want to talk about entities called data generation processes, where this is just, there is some sequence of events, there's some black box that produces a set of compounds that we then take into assays and gather information about. And the reason I want to start with this abstraction is because I think that at a very high level, this is essentially the iterative process that we go through in drug discovery. There is something that happens, whether it involves chemists or machines, and at the end of it, out come molecules that we're interested in testing. And they've been selected by lots of different decisions along the way. Here are just a few of those decisions that need to be made. We have to gather training data if we're going to build a model, for instance. We have to choose our input representation. We have to decide what kind of model we want to build. We have to actually train the model, and hopefully we have some held out data we can use to tune the hyperparameters and make sure it's doing something reasonable. Then we have to make predictions on the universe of molecules that we care about, whether this comes from a commercially available library or something that our chemists have put together in terms of the list of things that might be interesting and we need to prioritize. We have to make predictions on that set. Often there's post-prediction filtering that happens where you say, oh, I don't really like this one for reason X or Y or Z. And eventually you have to decide on the set of molecules that you actually want to pursue. Making or buying those molecules is an entirely separate challenge with the complexities of synthesis and even the vagaries of dealing with FedEx shipping your compounds from a vendor. And finally, you can test these molecules in an assay. I understand completely that assay development and assay cascades are not trivial topics, but for the sake of this conversation, I'm going to assume that this is just a switch we can flip. Now, I want to dive into the distinctions between retrospective and prospective validation here. And if this is the full data generation process, if this is what happens in a real world production setting of our models that we're building, I want to emphasize and make very clear that retrospective validation does not account for several of these steps. In particular, the set of candidate molecules that the model is evaluated on, the choice of filtering and ranking, and the process of actually acquiring these compounds. So let me give you a few very concrete examples, and these are contrived hypotheticals, but I hope you'll see the lesson I'm trying to convey. So let's imagine that this set of molecules is the full universe of things that we might consider. And when we're working in a retrospective setting, let's say we're building a model and we're using data from Kemble to validate it. There was some data generation process that happened, possibly in many labs, that resulted in a set of annotated compounds in Kemble. And maybe it's just this much smaller set of the total universe of compounds. However, what we don't know when we do our retrospective validation is how the model would have performed if it had been able to see these compounds too. We have a measurement of how well it did at matching the labels for these particular compounds, but maybe the model that we trained really would have liked these types of molecules better, and it would have put them at the very top of its rank list, and we would have never ever chosen these molecules in a real practical world setting. If I illustrate this with a particular selection, let's imagine that our model really liked these molecules down here, as well as one of the molecules that ended up in the retrospective set, and it did a terrible job. The model actually failed to predict that these compounds were going to be bad, even though it did really well on this retrospective set. Now, oftentimes I hear that there are some very good reasons why this might be the case. For instance, if we try to apply the model to a different chemical space outside the applicability domain, or if we're using slightly different filtering criteria, things like this. But the point is that those are not necessarily full explanations for this. The folks who generated the data in Kemble got to see all of this data, and they chose for some reason not to select these compounds, but our model chose them. And that means that we failed to see the blind spot that our model had because our retrospective validation was focused only on this limited set of data. Here's another specific example. Let's imagine that among the full universe of compounds, the data that we found in Kemble represents just this set. And not only, before we were sort of assuming that whatever model the folks in Kemble were using, whether that was a machine learning model or a chemist, because chemists are models too, whatever model they were using maybe didn't like all of these compounds, and so they weren't chosen. But we also have to consider the things that happen after the model makes its choices. Often you'll have a chemist sit down and look at things, or you'll have certain other criteria based on properties of the molecules that whittle down the set of compounds that you could choose from, regardless of what the model has to say about them. So maybe some compounds are too expensive or likely to interfere with the assay. perhaps that particular chemist that was making those choices had a gut feeling and decided that that molecule should not be pursued. And then what we ended up with for our retrospective validation is this limited set of compounds here. Well, now, if we're trying to validate a new type of model against this retrospective set, we're going to get some good data about these compounds, but we don't know how the model was going to look at these other ones. Let's imagine that all of a sudden our filtering criteria change. We no longer have a gut feeling about this particular molecule because now we're looking at the whole universe and we have a different chemist on board this week. And this molecule that was previously too expensive all of a sudden became cheaper. And so now it's something we can consider. Well, if these molecules end up being negative, we might consider this a failure in a production setting. But what really happened is that we failed to compensate for the fact that our filtering criteria were different. The models could be exactly the same, but because of what happens after the model makes its predictions in a real world setting, we get totally different results that can't be predicted by the retrospective validation. So that's why it's important to consider all of these steps of the data generation process. Here's another very abstract way of thinking about this that I think is helpful. Let's imagine there's two sets of compounds. One of those compounds is chosen by some process or maybe a set of processes. Maybe it's the set of compounds that you find in Chembl for the target that you're modeling. And the other set of compounds are the set of compounds that would have been chosen from that same chemical space in a prospective validation of your model. And these don't necessarily intersect that much. In fact, I think it's fair to say that the more they intersect, the more applicable your retrospective validation is going to be. If the set of compounds that happens to be in Chembl is the same set of compounds that your model would have chosen to evaluate, then your retrospective validation is going to be very good at predicting future performance because the sets of compounds that each method chooses are going to be very similar. But if they're very different, retrospective validation is not going to be a good estimate of future performance. So I think another way to say this is that prospective validation of a model is actually a comparison of a full data generation process with another process. There's some process that generates molecules with labels, like I showed you at the beginning, and some other process, and we want to compare them. The only levers we have to compare are the metrics we compute from the compounds that were selected, things like hit rates and potency, and these might be on totally different sets of molecules. But if we want to say that the difference in performance is due to a model, we have to make sure that we control for all of the other parts of the data generation process. Is the post-hoc filtering the same? Is the set of training data the same? Is the universe of molecules that each process had to choose from the same? If they're not the same, then the differences between these processes could be totally due to some difference in filtering or some difference in the set of candidate molecules and might have nothing at all to do with the model. And we just wouldn't know that unless we went through the effort of actually comparing these processes directly. So a couple sort of summary slides with a lot of text. Apologies. I want to make sure that it's clear that I'm not saying we shouldn't be doing retrospective validation. That's not at all the case. It's still very useful. It's much less expensive and time consuming than prospective validation, but we have to do it with an awareness of the biases that exist. We have to recognize that the data that we have in a retrospective setting was generated by some process that we usually don't completely understand and is certainly not going to be reproducible. If we ask that same lab who generated the data for a particular experiment in Chembl to do it again, would they get the same molecules? Maybe not. And we have to consider that level of variability when we're thinking about whether we're measuring future performance. As much as possible, your validation should try to incorporate some of these steps and decisions that happen before and after the model is actually involved. Make sure you think about the controls you're putting in place around filtering and the set of candidate molecules. There's lots of strategies that have been developed to help improve the robustness of retrospective validation. We all know that random cross-validation is just not very useful. And there's a lot of good strategies that have been developed as well for directly reducing bias, starting with things like maximum unbiased validation, which had its own problems, but there's been lots of follow-ups to try to make this better. I'll make a note which I hope isn't controversial, but I think is misunderstood. that if you do time split cross-validation, where you have a bunch of historical data and you have timestamps for each data point, and you train your model on the stuff that happened up until 6 months ago and then test it on the stuff that happened for the last 6 months, that is a retrospective validation. It is not prospective, because you have no control over the set of molecules that were chosen in that last 6 months. Your model has no skin in the game. You are relying completely on the process that was used to select those molecules, which your model has nothing to do with. Finally, one possible suggestion here to try to quantify the level of bias that exists is take a large unlabeled set of compounds, like download the enamine real database and run your models on them, and just look at the rankings. Even though you don't have labels, if the rankings are completely different, that's probably a pretty good signal that your models are going to select different molecules, and therefore comparing them in a retrospective setting is going to be difficult. And I would encourage us all to adopt a prospective perspective. I hope the alliteration is useful in encouraging this kind of behavior. So we have to remember that the retrospective methods that we use, while they can be useful, they only tell us how our model performs in isolation. If the model had no influence at all on deciding what compounds were actually selected, they don't tell us how they would perform in a real-world application. Remember that prospective validation is process validation. We need to consider what happens before and after the model is involved if we want to really compare these processes. One thing that can really help the robustness of a data generation process is to think about how you can systematize and automate different phases of it. You know, the first argument, of course, is, well, what about chemical intuition and the role that plays in our processes? And you know, I totally understand. Chemical intuition is very valuable, and it can be very helpful. And the point I want to make is that as we move into a world where machine learning and machine learning decisions play a larger role, the only way to take off the training wheels of those models and let them actually become better at what they do is to automate and systematize. If you have a chemist holding the hand of a model the entire time, it's never going to get better. And so there's a trade-off we need to make between performance now in the short term and performance gains over the long term. And as final food for thought, you know, chemists are models too. They also represent a data generation process that takes in information and outputs selections of compounds. And I don't think it's heretical to consider evaluating chemists the same way we evaluate models, and think about whether we can learn from that to do a better job of evaluating our models too. So that's the end of this first section. I saw there was a couple of questions here, but I think I'm going to stick to what we said at the beginning and go to the end before we go to the questions. So with this as a background, I want to share with you some very concrete results that we've obtained in applying machine learning to DNA-encoded libraries. And I think that what I just showed is a good background for this, because as far as I'm aware, the work I'm about to present is the largest prospective validation of a machine learning model in our field that's ever been done. There were about 2,000 compounds selected and purchased and tested for this particular application. And as I talk about the procedure and the science that we actually did, I'm also going to pause and be very clear about the data generation process and the types of decisions that were being made in that process, so you can see and judge for yourself how useful or prospective the work that we did was. So hopefully many of you are familiar with DNA-encoded libraries. Basic idea here is that by combining different pools of building blocks, you can very rapidly combinatorially generate huge libraries with lots of different molecules in them. Each molecule has a piece of DNA attached to it that codes for the identities of the building blocks in each of the positions. This allows for easy just DNA sequencing to find out the identity of a molecule. There's lots of details here about, you know, how often the DNA tag is actually faithfully representing what's on the business end. But this is a useful schematic anyway. If you take this library and incubate it with a protein that you're interested in finding binders against, you can wash off the stuff that doesn't bind and then elute the stuff that did bind and sequence it. You get sequence counts for each of these different molecules. Here I'm representing a 3-cycle library where the instances each have 3 building blocks. Now, if we're thinking about applying this in a machine learning setting, these individual counts don't contribute that much information individually. There's a lot of noise in the sequencing. Depending on how deeply you sequence, you might have an undersampling problem. And so one of the steps that we took for our particular application here with XChem and Zebi was what we call disynthon aggregation, where we essentially average over different positions in the molecules to arrive at a collection of disynthon building blocks. Now, these disynthons don't actually exist in the library. In particular, when we're averaging over like the B position here and we couple A and C together, that structure really doesn't exist in the library, but this is a useful aggregation in order to improve the signal to noise ratio. After we do this aggregation, we can assign labels to these disynthons and then use that to build a model. Now, the model that we built here, we trained a couple of different models, and we trained a random forest model that was actually trained at XChem using their expertise with working with this type of data historically, and we compared that to a graph convolutional neural network that was developed by our team at Google back in 2015. And just as a very quick overview of kind of conceptually what the graph convolutional network is doing, rather than working on a molecular fingerprint like ECFP or something like this that we used to train the random forest models, the graph convolutional neural network actually takes in the graphical structure of the small molecule as input, and then does a series of operations to produce a new representation of that molecule that happens to be a vector, just like your Morgan fingerprint. So in a way, the graph convolutional neural network is kind of a fingerprint learning machine that learns a representation that we hope is better than ECFP for actually making predictions about activity against the target that we're interested in. Now, after we trained this model, we downloaded large virtual libraries like MQL. We also had some virtual libraries that were composed of things that the folks at XChem could make easily using the building blocks that they had on hand. And our model made predictions for each of these compounds, and then we ranked them and went through a selection process to choose a set of molecules for experimental validation. And as I mentioned, we ended up purchasing nearly 2,000 molecules across 3 different targets for this validation. So now, before I show you the results, which I'm sure you're excited to see, I want to just pause and say, okay, here is the data generation process itself. So first, we had to gather the training data. I talked about this disynthon aggregation. The GCNN, the graph convolutional neural network, and the random forest use slightly different representations of the labels themselves, just because the random forest was designed for work with a binary classification, and GCNN we had 5 different classes based on whether things we thought would be non-specific binders or other ways of being negative. The input representation, as I mentioned, for random forest we use Morgan fingerprints. For the GCNN we're using these atom and bond features, and then the models themselves, we chose to directly compare these 2 types of models for this evaluation. Now, as we go into the stuff that happens after we decide on how we're going to build the model, we start to see some interesting things come up relative to what I talked about just a moment ago in perspective validation. So the models had slightly different training schemes. The GCNN saw all of the data, and we used oversampling to upweight the classes that had fewer examples. Because there was so many negative examples, they would have totally overwhelmed the positives. A similar thing was done for random forest, except overall the random forest ended up seeing less data just because of the infrastructure capacity type issues around shoving all of the data into a random forest model. As I mentioned, we made predictions on these different virtual libraries. And to be very clear, both the random forest and the GCNN were able to saw the same set of candidate molecules. So there's no difference there in terms of the comparison. After the model had made these predictions, there was a series of filters that were applied. First, there were property filters, things like molecular weight. We just really don't want to buy things that are too big. Then there's also this really interesting statement that I want to highlight. There was a non-systematic filtering done by chemists, but rather than just having them sit down and say, I would like to buy this one or I don't like this one, we asked them very specifically to restrict their filtering to molecules that were either going to be unstable or interfere with the assay. And you might say, well, why didn't you just automate that? And of course, that would have been a possibility. At the time, that wasn't practical for our collaboration, and this was. And so you'll see that we have some evidence that this was actually a fairly objective set of filtering, because we ended up with some really ugly molecules that I think any chemist worth their salt would have said, don't buy this, but we had specifically asked them not to think about things in those terms and only to think about them in terms of instability or reactivity. We then bought and tested those molecules, and now I will show you the results. So we trained models on 3 different targets: SEH, ER alpha, and c-Kit. The two rows for each target represent the hit rates for the GCNN and the random forest, and the colors indicate the potency of the different hits that we found. Just as a detail, everything was tested in a primary assay at a fixed concentration first, and compounds that showed good potency there were put into a full IC50 dose response curve assay. And the black bar here represents the 1 micromolar threshold. Everything to the left of these black bars had 1 micromolar or better potency. So right away, you see that on SEH, which by many would be considered an easy target, the GCNN was getting a hit rate of over 70%, and a hit rate of over 30% if you impose a 1 micromolar threshold, whereas the random forest had significantly less overall hit rate, both in terms of overall hits and 1 micromolar hits. Similar stories were seen for the other 2 targets, with progressing difficulty in terms of overall hit rates that we observed. And I think by any account, hit rates of this magnitude on any of these targets should be considered a grand success. This is much better than you would get from random high throughput screening or something like this. And the differences between the GCNN and the random forest are also very clear in terms of the number of hits that we found and the potency of those hits. If we dive a little deeper into these hit rates, these plots here show potency on the y-axis and similarity to the nearest neighbor in the training set on the x-axis. Here we were really trying to dig into the question of whether our models were actually generalizing. I'll throw in a couple of helpful guides here. So this threshold represents a 1 micromolar cutoff. Everything below this threshold is better than 1 micromolar potency. And the vertical threshold is 0.4 in ECFP6 Tanimoto similarity, which I think is a handy rule of thumb for deciding whether something would be considered by a chemist to be structurally unique or novel. And in all of these cases, we see hits in this quadrant where we have things that are novel and high potency. And if you're a little less concerned about overall potency, you can see that just to the left of this vertical line, we see lots of hits for each target. This band up here at the top is representing things that were tested in the single point assay and showed activity, but not good enough to move into the full dose response assay. So if we ask the question about whether these compounds are novel, I'm not going to spend too much time on these slides. I think that it would be great for those of you who are used to staring at molecules to spend a few minutes staring at these and asking yourself the question whether these compounds are novel or significantly different or not. In this first table here, we're showing hits that were found in our particular procedure compared to hits for these targets that have previously been reported in the Chemical database. And at least to my eyes and to many of the chemists that I've asked about these data, some of these hits are definitely things that would not have necessarily been found by just one or two steps away from things that were known previously. I'll further emphasize that if we look at rule of 5 type violations here, both SEH and CKIT tend to have things in sort of the golden range of log P and molecular weight. ER alpha tends to like big greasy things and has a lot more things with more than one rule of 5 violation. If we look at the similarity to things that were in the training set, this again gives us some estimate of how we're doing it as far as generalizing away from the training data. We have additional pairs of compounds where here are the hits that we found in our experiments compared to things that were in the training set for these different models. And again, we see some very significant differences. We also see some definitely conserved motifs and things that indicate that the model is learning particular substructures. Also for ER alpha, again, big greasy things are what it tends to like. And CKIT. And I'll make these slides available afterward for people that want to look more closely at this. Finally, I think it's important to consider the cost versus a traditional DEL follow-up. Pat Riley put together this table, which I think is very illuminating in terms of in a traditional DEL follow-up, you'd run your DEL selection, you would look at the enrichment scores and then actually decide, okay, here's a few molecules we want to follow up on. You'd make them off tag, which can be fairly complicated because it's a totally different synthetic process than the library building itself. And let's say you decided to follow up on 75 different compounds, you'd end up with a total cost somewhere between $100,000 and $300,000 for that particular follow-up. In the work that we did here, we spent a trivial amount of money building these models and evaluating them on the targets that we trained. Of course, we're not including FTE costs of the engineers that were involved necessarily, but now that we've worked this out, it's fairly marginal. The number of compounds acquired was much larger than you might do in a traditional DEL follow-up, and the total cost ends up being, many factors smaller than what would happen in a traditional DEL. And as a result of the work that we've done here and the success that we've seen with applying these models to DEL data, our partner Zebi AI has launched an initiative that we call the Chemome Initiative, where here the goal is really to find chemical probes for many, many different protein targets. Just a small fraction of the protein targets in the human proteome have chemical probes that can be used for starting points for drug discovery and other applications in systems biology, et cetera. And so what we're doing is partnering with academics and other nonprofit groups like the Structural Genomics Consortium and running DEL selections against their targets and then building machine learning models and suggesting hits from commercially available libraries, and then sending those to the partners for testing to actually identify these new chemical probes. And the goal is to really scale this up into hundreds and thousands of targets over the next few years. We're making very good progress on that now. And you can learn a lot more about that here at their website, zebiai.com/chemome. So trying to stay on time, I'm going to spend about 10 minutes or so talking about the open reaction database. Now, the way that this fits into the world of everything we've been talking about so far, I talked about prospective versus retrospective validation, and I talked about a particular example of prospective validation of these different models. And one thing that we haven't spent a whole lot of time, at least in this talk, talking about is where the data actually comes from and ways we can work together to generate the data that we need to build better models. And the open reaction database is really designed to fill a couple of those roles in the realm of reaction prediction and planning. So as I showed before, this process of make, design, and test. This is an idealized representation where each of these stages is given approximately equal weight. In reality, we all know the process looks a little more like this. And especially, I want to emphasize that synthesis itself can take a huge amount of time and resources. In fact, I would claim that it is the major bottleneck in the work that we do in terms of active learning and lead optimization. And so machine learning efforts are underway and have been for many years in order to improve our ability to both plan reactions and predict their outcomes, and just in general make this process more effective and efficient, so that when you decide to make something, you have confidence you can make it, and when you try to make it, it works the first time. Now, this really came out of a number of different meetings that started in the automated synthesis community. And I remember being at a DARPA meeting a couple of years ago now, where I heard a lot of people talking about automated chemistry platforms and saying things like, there's a huge amount of data that exists and we have nowhere to put it. And I said, well, Google's pretty good at deciding where to put data. And so we decided to get together and put together this effort. And we've been working on it for a little over a year, and I'm really pleased with the progress we've made, and I hope you'll see that as well. So the overall goal is to support machine learning and other downstream efforts in reaction prediction and planning. And the way that we're doing that is by building a database as well as a structured schema for holding reaction data. And we want to make sure that we make it easy for people to browse and interface and submit data to this database. We want it to be freely and publicly available for anyone to use. I think a lot of people consider reaction data, at least some of it, to be pre-competitive. And we want to leverage that and make sure that this effort is totally open, both in terms of the code that is used as well as the data itself. Now, we have a few non-goals here. We are not trying to be the one-stop shop for machine learning in this area. We are trying to be the one-stop shop for data that is used in machine learning applications. So we're not going to be building these models ourselves and trying to say, here's the best model in the world. We're very limited in our scope to focus on the data itself. We're focused less on sort of describing things in a way that could be used for automated synthesis than we are in describing what the chemist actually did when they ran this reaction and how things were actually set up and observations along the way. And we don't currently have plans to support a structured database of NMR data or mass spec data. All of these types of data can be attached and included in the schema in unstructured ways, but we're not trying to reinvent the wheel in terms of how that data itself is stored and represented and shared. We've assembled a fantastic governing committee composed of people from lots of different industry and academic segments. In particular, Connor Coley and I have been working very closely on the infrastructure development and the submission workflow that you'll see in a moment. We've also built a fantastic advisory board that we feel really represents a lot of the people who are generating this kind of data. And our real goal and hope is that by working with these people, we'll be able to make the data generation process itself more systematic in terms of data being encoded in a digital format from the start, and in a way that can be searched and shared with the rest of the world. The primary use cases here are high throughput experimentation, where often you'll run a factorial experiment where you'll maintain many things as constant, but have a few variables like the types of inputs or solvents that you might be using. And then we've made it very easy for people to define template reactions that represent one instance of that factorial experiment, and then pair that with a spreadsheet to very quickly enumerate a large data set of reactions. We also support traditional bench chemistry, where we put a lot of effort into an interactive web editor that allows people to go in and put in data, whatever data they have, to build an instance of our schema and then submit it to the database, as well as submit it along with their papers as part of supporting information that can be shared freely as a digital format, rather than having to try to parse these kinds of details from, say, a PDF. The schema itself is organized by reactions and has many different components. The underlying schema is fairly comprehensive. We feel that it has the capacity to capture nearly any kind of data that you might have when you're running these reactions, much of that in a structured way. But the point I want to emphasize here is that not all of this data is available for every reaction, and we're totally aware of that and comfortable with accepting whatever data happens to exist. And we have certain validations and requirements that we use to guarantee consistency and interoperability and things like this. But in general, we're trying to take the data in as raw a format as we can and structure it, and then share that with the world without imposing our own biases or filters on types of data quality and things like this that are going to be important for deciding what goes into the actual models. The underlying schema technology is built on protocol buffers. If you're not familiar with this tech, a quick Google search will show you that this is a Google technology that it's the primary way that we store and transport data within Google. There's also lots of external applications of the technology. It's smaller, faster, and simpler than XML. If you've ever had to deal with parsing XML and then you give protocol buffers a try, I think you'll find them refreshing, in particular because protocol buffers can be compiled into language-specific classes, like this Python class for defining the mass of a thing that is very easy to use. I mentioned we built an interactive editor here with lots of different pieces. This is continually evolving in terms of the UI and UX. And we're hoping to hire someone who actually knows what they're doing in that space to make this really pretty and even more functional than it is now. But it serves its purpose now and has things like an interactive compound editor for drawing structures, as well as auto-generated reaction summaries based on the information that the user puts in to define their reactions. And one thing I should mention here is that from the perspective of generating data, not only gathering data that happens to exist now, one of the hopes that we have here is that with this great advisory board we've put together, and as we start to see this data come in from lots of different sources, that'll be an opportunity for us to look at the data that exists and say, we're missing information about this particular reaction type that would be really helpful in improving the state of the art in terms of the models that we have today. Why don't we get together and put out a request for data to be generated of this particular kind? It's kind of a new way to think about data generation rather than sort of the siloed way that it happens in a lot of the drug discovery companies and other settings, to think about working together as a group to identify the data that needs to be generated and then making that happen to improve the models and improve the sort of, lift the state of the art in terms of the modeling that can be done here. The database is entirely open. The code and data are hosted on GitHub. The submission process itself is a pull request to a repository on GitHub. So everything is done out in the open, along with appropriate licenses. So the code itself is available under an Apache 2.0 license, which is a very common open source code license. The data itself is under a Creative Commons license with a share-alike clause. And we also have some terms of use that were drafted in kind by some friends at Google. And these terms include things like, acknowledging that you have the right to upload the data in the first place and things like that that are not covered by the license terms themselves. Our roadmap is, as you see here, we started working on this in October of 2019. In September of last year, we began alpha testing with a small group of individuals to really kind of battle test and work out the kinks in both the schema itself and the submission pipeline. We made some really good changes to the schema just before Christmas. And we're now in an open beta phase, preparing for a public launch near the end of Q1. We're really excited about this effort, and we're trying to scale it up to get as much data in there as possible before the formal launch, along with tutorials about how to use it in downstream applications and other things like that. We'd love additional contributors, both to the data itself, as well as the code base or UX or whatever, in whatever way that you think you could be useful. We are happy to have you on board. And with that, I'll conclude and just say that there are a ton of people that have been involved in this work, both at Google, as well as XChem and Zebi. And in particular, I've been working, as I mentioned, very closely with Connor Coley and others on the Open Reaction database. And we want to give special thanks to our alpha testers who helped us to make some important changes there. There's a lot of people I didn't have room to show on this slide or couldn't find pictures for. Very grateful to be working in this field at this time where so many exciting things are happening. And I'm excited to take your questions now.
Thank you. That was a great talk. So I'm just going to go right into the questions. There are a lot of questions about the differences between the graph convolution and the random forests. There's basically a simple question is, do you have some understanding of why there's a performance difference?
Yeah, that's a great question. So I think I want to go back to this slide and emphasize that the random forest is trained on these Morgan fingerprints, which are extremely useful and powerful substructure, presence and absence identifiers. And the only issue with that representation is that it was designed to be a general representation of molecules useful for many, and it ends up being useful for many different tasks. But the graph convolutional neural network is able to learn essentially a better fingerprint than Morgan fingerprints for this particular task of predicting activity against SEH or CKID or ER alpha. And so I think the core difference is just that the model itself is able to take in the raw chemical graph structure and see millions of data points from the DEL selection and use that to learn a better representation than Morgan fingerprints for this classification task. Now, of course, as I mentioned, there are lots of details in terms of exactly how the models were trained. But this is one of the first applications of graph convolutional neural networks where we've seen a significant difference between the Morgan fingerprint world. And I think that has largely to do with the fact that we're training on so much more data than we ever have before. We have literally millions of data points going into these models, and that is allowing the graph-based representations to pull away from the more traditional fingerprint representations that have been the workhorse for so many years.
And you don't think increasing the bagging or the number of trees or depth is going to be enough for a random forest to be able to capture the information as well? Or is it just not practical?
That's a great question. I do not know how much hyperparameter optimization was done at XChem to build these models. However, we allowed them to build the models as partly to avoid a straw man situation where we would just build a random forest baseline at Google and compare it against our big powerful machine. So we have some confidence that they built the best models that they knew how to build, and maybe a traditional model will catch up eventually with some particular tuning or framing. But in this particular comparison, we saw a drastic difference, and it's not clear to me that just tuning the parameters of the random forest is going to overcome that.
So another question that is related is the chemist removal of molecules. So do you know the process of those compounds were tagged? And if so, could you build a model around that?
Sorry, could you say it again?
This is how chemists were removing models from the data set. So the question is, do you know the criteria used, and could you build a model around that?
Yeah, great question. We spent a huge, inordinate is probably the right word, inordinate amount of time deciding how to phrase the filtering that was done here, because we care so much about the systematic application of these models. And so the way that we wrote it in the paper was that the filtering was done by automated or automatable filters. And so, yes, I believe that if we sat down with these chemists, we could write a set of criteria and algorithms that would approximate at least the filtering that was done by them. It's not going to be perfect because, again, it was human chemists sitting down and looking at things, but I think we could get to a place that approximated that very well.
And Brian Cole asks if diversity is a criteria used in your validation. Why isn't that part of the selection step during the post-filtering process?
Sorry. Yes, it is. I left that out. We used a sphere exclusion algorithm to make sure that the molecules we were actually buying were not just analogs of each other.
And so Woody Sherman asked, why did you choose such easy targets?
On this one, I claim no responsibility. We at Google, we were actually totally blinded to the targets until after we had purchased and tested the molecules. But I'm also not just putting the blame on XChem necessarily. This was really an exploratory project. We were trying to see if this was even feasible to build these types of models on this kind of data. And so we started with SEH, which we knew was an easy target, and then we moved to progressively harder targets. The chemo effort that we're running now is really the battle test of what you brought up, Woody, because we're currently running on targets that are much more difficult. They may not even have known competitive inhibitors that we can use to aid in the selections. And stay tuned for the results of those experiments in the future.
So following up with that in his question, I'm assuming you looked at the standard similarity-based metrics of doing rank ordering and things like that. And when you don't have competitive information, you just can't do that anymore. Is that going to be a feature of how you analyze the next round of results, or have you done that already?
Yeah, great question. So I'll mention an experiment that I didn't mention here, which is that in the paper, we actually report on a purely similarity-based selection of compounds based on hits from the DEL selections that ended up yielding no hits whatsoever. So we definitely did some preliminary work in that area of just exploring the similarity-based thing. But things do get more complicated when you don't have a competitive inhibitor, because you can't then distinguish between things that bind and inhibit versus things that just bind. So there's no functional readout of the DEL selection itself. So that definitely has, we expect some effects on hit rates and overall performance, but I don't have anything to share publicly about those results yet.
I'm going to paraphrase the next question a little bit from Dominic, but he's making the point that it depends on the therapeutic need whether basically the range of potency is important. So if you have weaker hits but lots of them, sometimes a therapeutic target is better associated with that. And sometimes the stronger hits are a little bit less useful, especially if there's other side effect issues or they hit a bunch of kinase panels. So is there any thought about how you can look at the history of the therapeutic target and how you're designing what exactly you want to get out of the prospective system?
Yeah, that's a fantastic question. And I think that highlights that what we've shown here is really just scratching the surface. I think there's a whole bunch of additional optimizations that could be done in terms of both the model architectures and the types of things that you're bringing up here with, focusing on exactly what is needed for the target, the exact types of chemical space you want to avoid potentially when you're making your predictions and working on things. And for the chemo effort, we're particularly focused on finding chemical probes, which tend to have at least some of a common profile in terms of you need something with a potency of about X and certain chemical properties in order to be useful as even a starting point for the types of work that you might want to do to find real hits and then turn them into leads. And so we're focusing on that problem now, where we're generally trying to find at least a few sub-micromolar hits for every target, and that's just the particular setting we're working in, but it may not be right for every program.
And two switching topics to reactions. Two last questions from Lee Herman and Colton Hicks is: one, do you have failed reactions? And two, do you allow theoretical reaction data? Great
questions. So the schema is designed to accommodate negative reactions in a couple of ways. One is that let's say what product you were going for. You can put in a desired product and say you got no yield. You could also carry over materials from the starting materials into the products of the reaction. So the schema itself totally supports that. And we're very interested in making this a place where people can put negative reaction data and receive credit for donating that data to the community, because we know that's super important for building these kinds of models. As far as theoretical reactions go, the schema is really designed for reporting experimental conditions. We do have space in there for things like computed and calculated features that might be used in downstream applications. Like Abby Doyle has done a lot of work in this area with quantum mechanical descriptors built on top of reactions. But It's not currently designed. Like I think it's possible to put theoretical reactions into the schema, but we're not really encouraging that kind of contribution now because it's not necessarily going to be useful for the applications we're most interested in.
All right. Well, once again, thank you very much. That was a great talk, and I look forward to your follow-up paper.
Great. Thanks so much for your time, and please feel free to email me at kearnes@google.com if you have more questions or follow-up.
Yep, and the video will be up in a day or two once we get it all analyzed and put up on the website. So thanks everyone, and let's thank Steve virtually. Thank you.
Bye.